Good morning. My name is Hannah Lott from Product Development, and in today's presentation, I'll be focusing on an antibiotic, Sultamicillin, detailing how the drug is made and the regulatory context in which the drug exists. Certain drugs are indispensable to the pharmacopoeia of modern medicine. One such class, perhaps the most significant, are antibiotics. Industrially produced since the 1940s, antibiotics are extensively employed by clinicians, both therapeutically and prophylactically. Global antibiotic sales alone generate $42 billion annually and account for 5% of all medication prescribed. The most extensively utilized antibiotics are the beta-lactams, a family which includes penicillin, methicillin, and ampicillin. Given these blockbuster drugs are no longer patent protected, it is in the economic interest of prospective manufacturers, such as yourselves, to investigate the commercial viability of the bioprocess by which these valuable compounds are made. Firstly, let's take a brief look at the chemistry of sultamicillin and discover how that informs the methods used to produce this compound. Sultamicillin is what's known as a semi-synthetic antibiotic, chemically derived from the naturally produced penicillin. The bactericidal activity exhibited by sultamicillin is due to the presence of ampicillin, a molecule which consists of a core penicillin nucleus bonded to a synthetically introduced side chain. Sultamicillin is also classified as a beta-lactam antibiotic due to it containing a beta-lactam ring, a chemical moiety conserved in all penicillin derivatives. However, due to this ring structure, all beta-lactams are rendered vulnerable to enzymatic degradation by bacterial beta-lactamase. To negate this resistance mechanism, ampicillin is formulated with sulbactam, a beta-lactamase inhibitor, resulting in the far more effective sultamicillin. The bioprocess by which sultamicillin is produced was originally proprietary information, with the drug patented by Pfizer in 1989 as Unison. However, Pfizer no longer has exclusive rights to manufacture sultamicillin as the patent has expired. As such, the manufacturing process is publicly available knowledge. Although attempts were made to obtain this information directly from Pfizer, the following details had to be collated from articles and handbooks published by industry stakeholders. While an expired patent does create potential for market saturation, there remains little competition as other Australian-based pharmaceutical companies are yet to manufacture sultamicillin. Therefore, the domestic market is ripe for a homemade generic version of the antibiotic. The production of sultamicillin can be broken into 3 stages. Firstly is upstream processing, which encompasses strain selection and development, inoculum maintenance, expansion and storage, and finally substrate preparation. Secondly is the bioprocess itself, which involves fermentation of the producer microorganism Penicillium chrysogenum. At this stage of production, bioreactor conditions are continuously monitored to maintain optimal growth and product formation kinetics. Following product formation, the compound of interest is recovered from the fermentation broth. Then follows a series of procedures which increasingly purify the isolated compounds. The antibiotic is then formulated and packaged, ready for human consumption. Perhaps the most important aspect of upstream processing is strain improvement of the producer organism. In the case of sultamicillin, this has already been performed over the preceding 80 years on the filamentous fungi Penicillium chrysogenum via conventional mutagenesis and more recently genetic engineering. The industrial strain of choice is Wisconsin 541255, developed for its consistently high titers of penicillin G, the precursor compound from which sultamicillin is derived. In submerged culture, this strain forms pellets as opposed to long filaments, which has the advantage of reducing the viscosity of the broth and improving bulk oxygen transfer. This organism is generally recognized as safe and poses no threat to the health of plant operators. Maintaining the fidelity of this inoculum is simple, with specific temperature and media requirements prescribed by the supplier, the American Type Culture Collection. Given antibiotics command a lower price than biologics, less pure, complex media is economically justified and is typically composed of cheap byproducts recovered from agricultural processes. Molasses is the favoured carbon source due to it containing the rate-limiting substrate of glucose in adequate quantities. Molasses constitutes 10% of the production media, whereas corn steep liquor is 4 to 5%, supplying nitrogen for incorporation into the mycelial biomass. Corn steep liquor also supplies phosphorus and an important side chain precursor, phenylethylamine. Also formulated into the medium is phenylacetic acid, a molecule that expedites the biosynthesis of penicillin. Vegetable oil is employed as an anti-foam agent and ammonium sulfate as an anti-lytic additive. The final step in upstream processing is the expansion of the P. chrysogenum inoculum. This is achieved using a seed chain encompassing 2 to 3 initial seed growth phases, beginning with the resuspension of lyophilized spores and culminating in the inoculation of the production fermenter. Next comes the fermentation itself. Bioreactor operational parameters are chosen according to the physiology of the producer organism and the biosynthetic pathway of the product. In our case, the product is a secondary metabolite that is subject to non-growth associated kinetics. Consequently, penicillin biosynthesis only takes place during P. chrysogenum stationary phase, when the net growth rate is 0. Because of this distinction between primary and secondary metabolism, the growth or trophophase and production or idiophase are physically segregated during fermentation, with each induced by different optimal conditions. Physical partitioning is achieved using a multi-stage system consisting of a minimum of 2 bioreactors. The first vessel is a cell mass production reactor, a stainless steel chemostat containing the submerged culture of penicillium, continuously fed with sugar and corn steep liquor, with the substrate supplied at the same rate at which it's consumed. This feed not only maintains pH, but also prevents substrate inhibition by excess glucose. In addition to prolonging the exponential growth phase, a fed batch reactor stretches a company's dollar, as it circumvents the inefficient use of substrate as seen in batch cultures, and realizes a higher cell density, which in turn facilitates product recovery. The broth is vigorously aerated with ring sparges and supplemented with oxygen to accelerate mycelial growth, culminating in a doubling time of 6 hours. Mechanical agitation is applied with radial impellers to not only ensure homogenization of the nutrient broth, but to also generate shear forces that serve to enforce a uniform mycelial pellet size and morphology. As a mesophile, penicillium's optimum temperature is maintained using cooling coils, designed to transfer the heat generated by rapid exponential growth. Once the bioreactor is 80% full, the trophophase ends, and the broth is transferred to a second chemostat for the induction of the idiophase. In this bioreactor, the flow of glucose and corn steep liquor is slowed, reducing cell growth and inducing a condition of starvation for penicillin production, whereas ammonium and phenylacetic acid are continuously fed for incorporation into the structure of penicillin G. All in all, the fermentation cycle takes from 120 to 200 hours. Before penicillin G can be enzymatically converted into the semisynthetic ampicillin and then chemically transformed into sultamicillin, the natural penicillin must be recovered from the fermentation broth during downstream processing. This downstream recovery process encompasses filtration, extraction, absorption, crystallization, and drying, in addition to formulation and packaging. Firstly, the mycelial mass is separated from the broth by high-capacity rotary drum filters operated under a vacuum. These mycelia are washed on the filter to elute the antibiotic and then disposed of. The resulting penicillin-rich filtrate is promptly cooled to 4 degrees to minimize chemical and enzymatic degradation. The pH of the broth is lowered to 2.5 via the addition of sulfuric acid. At this same temperature, solvent extraction is performed using continuous, countercurrent, multi-stage centrifugal extractors with butyl acetate as the organic solvent. Using Podbilniak extractors in series, this process can recover up to 99% of penicillin excreted. However, because penicillin only has a 15-minute half-life at this temperature and pH, extraction cycles are performed rapidly, typically in less than 90 seconds. Once extracted, the penicillin-rich butyl acetate phase is clarified using an activated carbon column. Penicillin is then back extracted into water by the addition of alkali potassium acetate and precipitated as a potassium salt of penicillin, with the spent solvent recovered for reuse. The crystals are purified via rotary filtration, polished with anhydrous butyl alcohol, and vacuum dried to produce crystalline potassium penicillin G. However, this is not the end of our story. Penicillin G needs to be enzymatically converted to ampicillin before formulation with sulbactam. This is achieved in aqueous solution using an enzyme cascade. Initially, immobilized penicillin G acylase, covalently attached to Cefidex beads, hydrolyzes penicillin G, producing 6-APA and phenylacetic acid. Then, amino ester hydrolase attaches an acyl side chain scavenged from D-phenylglycine amide to the 6-APA nucleus to produce ampicillin. Once the reactions are complete, the Cefidex beads are filtered, washed, and reused for up to 100 batches. The phenylacetic acid can also be recovered and reused in subsequent penicillin fermentations. The final step in the synthesis of sultamicillin is purely chemical in nature and is outlined in the patent Pfizer submitted 29 years ago. It entails the O-alkylation of ampicillin with a halomethyl ester derivative of sulbactam to produce the double ester of sultamicillin. Following this final modification, sultamicillin is precipitated and dried in vacuo to render the final product a dry powder for reconstitution. Because sultamicillin is degraded in acidic conditions, the antibiotic is only available as a parenteral formulation for either intravenous or intramuscular administration. It is packaged in single-dose glass vials containing either 1.5 or 3 grams of sterile powder for reconstitution. Alternatively, it is available in 15-gram pharmacy bulk package containing many single doses. Costs for sultamicillin production are estimated to be $10 per kilogram of glucose utilized as substrate, with raw materials and utilities the most expensive aspects of the bioprocess. The bulk of utility expenditure is put towards Sydney Water rates for discharged industrial effluent, as outlined in their acceptance standards. To cover the costs of treating trade waste, Sydney Water charges manufacturers according to the quality and content of their wastewater, in addition to application, consent, and inspection fees. Additional financial penalties are incurred if discharged substances exceed the published accepted standards, as outlined in this link. Therefore, it's in our economic interest to reuse as many raw materials as possible. Now that you understand the bioprocessing of sultamicillin, a note on its regulatory context. All pharmaceutical manufacturers in Australia are subject to Good Manufacturing Practice Guidelines, as outlined in the Therapeutic Goods Act. Compliance involves certification by the Therapeutic Goods Administration upon demonstration that quality is built into each batch of the product during all stages of the manufacturing process. Documentary evidence is required to ensure a high degree of assurance that processes will consistently produce a product meeting its predetermined specifications and quality characteristics. Quality assurance is even more important in this age of antimicrobial resistance, with evidence showing subtherapeutic levels of antibiotics promote increasingly virulent strains of bacteria. Antibiotics of poor quality create this subtherapeutic effect and may even result in treatment failure. Therefore, in accordance with antimicrobial stewardship, manufacturers are required to ensure the identity, dose, quality, and purity of their product so as to preserve the effectiveness of remaining antibiotics. I trust after today's presentation you will agree with me that sultamicillin represents a valuable addition to our company's product portfolio. That brings me to the end of today's podcast, which I believe demonstrates the technological feasibility of sultamicillin production given our company's existing infrastructure. If you have any outstanding questions or are interested in some further reading, I'd like to direct your attention towards the references listed at the end of the copy of your slides. Alternatively, if you see me wandering around the campus, you're more than welcome to flag me down and fire away. Thank you for listening, and I look forward to discussing this business opportunity further.